Thank you, Bobby. Our last speaker for the day, last but not least, is Dr. Issa Dahabreh. Dr. Issa Dahabreh is the section head for Epi and Data Science at the Smith Center and an associate professor of Epi and Biostats here at HSPH. He got his medical degree at the National University in Athens, an MS in Clinical and Translational Science at Tufts, and then his doctorate in Epi here at HSPH. I think that's, as you already figured out from the conversation so far, Issa, with his joint appointment at CAUSALab and at the Smith Center, is the glue that brought us together, this partnership together, and it is a major reason we're all here today. But one of the joys, I would say, of working with him is that he has the distinct ability to take everything back to first principles. Like you might ask him, what kind of salad do you want? And he has like first principles guiding him there. And so we're here for quite a treat. Issa, thank you for joining us today, both as our resident epidemiologist and philosopher at the Smith Center.
These are hidden slides from over there. Okay. So I'm Issa. I don't know what to say after that introduction. So I'm going to try to be a little conciliatory between the designs. And so I changed my title only slightly because I want to give a couple of examples where the trial alone is not enough, but maybe the observational data alone is also not enough. And then maybe you use both and you get something better. Okay, so we often, and in part of this discussion today, you saw this, think of trials and observational studies as mutually exclusive alternatives. And then I like one, I don't like the other, or we should do that one, we shouldn't do the other one, or I believe that one, I don't believe the other. And so I think it's better to start with a research question, and then you look what you have, and you do the best you can do to answer it with what you have. Sometimes it will be one of these two, sometimes it will be both, sometimes it will be a third thing. And so the argument I want to make via two very specific examples is that often you'll be called to combine information from diverse sources to answer the question in the best possible way. Now, there's a disclaimer needed. I did study medicine when I was a child. Didn't go very well for anyone. But I don't practice, and so you shouldn't take medical advice from epidemiologists. So I'll try to give the rationale for some of these interventions, but there's maybe 70 people in the audience who know better about the specifics. Okay, so with that disclaimer, my first example is going to be thrombus aspiration for myocardial infarction, ST elevation, myocardial infarction. And so the idea is you're trying to establish normal coronary blood flow. Bobby showed a similar picture before. And so people thought maybe you could put a device through the thrombus, and it has a little bit of suction on it. And so what will happen is you won't get these little things dislodging, and maybe it makes the stent placement a little easier. Again, don't hold me to the details, ask Bobby. Okay, but so the idea was maybe this thrombus aspiration kind of technology could be used to improve blood flow and lead to faster resolution of the ST elevation. And at the time, the trial I'm going to anchor on, which is called the TASTE trial, at the time the trial was designed and done, most studies before it had not focused at hard clinical outcomes. There was a smaller trial that had come out and suggested that maybe there is a survival benefit. And then there was some suspicion in the community that that benefit may come at the cost of increased risk for stroke. So people didn't know, and they thought they could do a very large registry-based trial. And so the trial happened in Sweden, pretty similar setup with a Swede PAD, I think. 29 Swedish centers and one Icelandic one. I won't be using the Icelandic one too much because it makes things a little more complex. And importantly, the trial used the powerful infrastructure in a registry that's now called the Sweetheart Registry, And so the trial happened entirely within the registry. The randomized individuals are a subset of the people in the registry, and outcome ascertainment was done via the registry. So this is a very point of care, routinely collected data supported, a large registry trial. And the treatments were manual thrombus aspiration, like in the picture before, plus PCI versus PCI alone. And there was some encouragement to use stenting in both groups, and then medical therapy was left at the discretion of the treating physician. So the randomization is for thrombus aspiration versus no. And I'll focus on the outcome of death from any cause. And so these were the results, the main results of the trial at 1 year in the New England Journal of Medicine. They had 11,000, this is the Swedish component, 11,600 eligible patients. Of those, about 60% were actually recruited into the trial. So about 40% said no, or the providers thought randomization was not a good idea. And those 7,000, but 60% was followed up for up to a year. And these are the risks of death at 1 year, and they're essentially the same, 5.3 and 5.6%. And so this was a influential trial. This is a secondary benefit, I guess, of the registry infrastructure, that you can see the use of the treatment before, during the trial, and after. And so it starts at a low baseline. There is a slope of increasing popularity, kind of flattens out. Then the trial comes out, the results of it become public, and then there is a precipitous decline, and then again a flattening. I guess these are the points, Eric. These are the people Eric was calling the supporters of a device. They'll keep using it after a negative trial. So this was an influential trial, but as is known for even very large pragmatic trials in cardiology, people were not convinced. They thought those 40% non-enrolled eligibles meant something. And so there was a concern about the representativeness of the trial. This is another area where the registry infrastructure comes to help, because it allows us to take the eligibles who were enrolled, take the eligibles who were not enrolled during the time the trial was recruiting, and compare them using methods similar to the ones you saw in the other talks, of trying to get a standardized mean difference for different variables. And so you can see there were some pretty sizable SMDs, Killip class being an important acute predictor of mortality, for example, was highly imbalanced, and all of these are well above the 0.1 threshold that people like to use. So lots of differences, and almost all of them suggesting that sicker people, more acutely sick people, were out of the trial, were not enrolled in the trial. So the question is, if these variables predict the magnitude of the treatment effect, and they have a different distribution between participants and non-participants, can we really apply the treatment effect of the trial directly to the entire target population, which here I'll take to be all the trial eligibles in Sweden during the period. And so one way to start examining this is to maybe build a predictive model for the probability of participation, again, using the registry infrastructure. And I've listed here a bunch of variables that I'm not going to read, but what you see is there is a distribution, and although it has a pronounced peak at roughly the prevalence of the probability of participation, it also has a pretty long tail. And so these probabilities are people who are relatively unlikely to be trial participants, even though they met all the trial eligibility criteria. And so the question is, if the answer in the trial is driven by the people here, what should we do, and how can we learn about the entire distribution, including the people down here? And so one way to pose this question is to say, what would be the effect of the randomized treatments studied in the trial, were they to be applied in the target population of all trial eligible individuals? And so the intuitive idea here is going to be, I will do some statistical magic to scale up the treatment, see what would happen had it been applied to the entire population. Statistical magic is a technical term. And so, I don't want to go into the technical details, but the intuition is that we will roughly view the trial as if it's a biased sample from the target population. And we have a sample of that target population in the registry. So the trial is nested within that sample. And then there is multiple ways, and I have a couple of references here if you want to see details. You can model the outcome and try to apply the trial result to the broader population, or you could use some kind of weighting method, which essentially treats trial participation as a treatment. So it's very similar to methods you saw before, except the outcome is no longer treatment. The propensity score is for participation in the trial, and the reweighting aims not to bring balance between treated groups, but between participants and non-participants. And so we did this, but I want to issue a caution that there are some very strong assumptions needed. So the first one is that the trial engagement itself does not affect the outcome, except through the assigned treatment. And so informally, this is an assumption that there is no Hawthorne effect. Maybe that's believable for all-cause mortality. And there are no effects of participation on adherence, no effects of participation on treatment via adherence. So I'm sorry for the typo. And so that one in pharmacotherapy perhaps would be a very significant assumption, a strong one. Here we're talking about essentially a point treatment over a small time period. The amount of non-adherence in the trial was very low, so perhaps it's okay for this application. The second one is that trial participants and non-participants are exchangeable. In some sense, there is no confounding of participation by unmeasured variables. So again, I'm kind of viewing participation as a treatment in itself, a special kind of treatment that doesn't affect the outcome except through assignment, but one that is also unconfounded. And then the third one is that perhaps not in the same proportion, but the covariate patterns in the target population have non-zero probability of being seen in the trial. An important assumption that is not needed, and that's why it's not listed here, is any kind of assumption that there is no confounding outside the trial. So I'm not making that assumption. And as such, I'm relying on different sets of assumptions compared to an observational study done outside the trial. So we don't assume no confounding outside the trial. So these are the results. The trial ones showed the relatively low risk compared to the transportability results. These are the transportability results from two different methods that estimate the exact same causal quantity. So you like to see agreement between this row and this row, and then any difference from the top row indicates, in a sense, our ability to adjust for the case mix in the trial versus the target population. And as we said in the beginning, the people in the target population were sicker, more acutely ill, and so you see the risk goes up after the adjustment. But the risk difference stays about the same, and the confidence interval always includes 0. So you could say, even though the participants in the trial were not that representative of the entire registry, the conclusions that we could have drawn about them are similar. Now, again, because the registry collected data before and after, as I showed you in that utilization graph, we were also able to do an observational analysis before and after. This is the combined results of observational analysis done before and after the trial, and they very much agree with the transportability results. So that's an indirect support that these results make sense, because as we said, the trial, the observational studies, and the transportability rely on different sets of assumptions. The target trial estimation from transportability and the observational analysis estimate essentially the same causal quantity, and so agreement is good to see here. I know the temptation is very strong in all of you to ask me if we could combine the transportability analysis that uses the trial outcomes with the observational studies before and after the trial, and we can, but I don't have time to tell you about that. Okay, so I think an interesting question is, can this be done in the United States? And in my telling of the story, the transportability analysis in TASTE benefit greatly from the existence of the registry infrastructure. So with United States data, more creativity is needed. And so very briefly, I'll go through an example for transcatheter aortic valve replacement. This is work from the people at the Smith Center. I had like a super minor consultative kind of involvement, so forgive me if I say anything that's a little off. But basically the idea is these are like marvels of medicine that happened after my time in medical school, where you go and you deliver a replacement valve as an alternative to open heart surgery, what we used to call it in my time. And then the prosthetic displaces and functionally replaces the native valve. And so there is two kinds. One is like self-expanding, and the other one needs a balloon. And the work is about the self-expanding one here. So amazingly, the research program for the development of that valve was done in a risk stratified way. So basically, there was an extreme risk study that was observational and compared surgery versus medical therapy, TAVR, this new intervention versus medical therapy. And then there was a high risk and a lower risk group. And these two were randomized trials against surgery. So it's always TAVR. In the extreme high risk is TAVR versus medical. In the high risk and intermediate, if you will, risk, it's TAVR versus surgical. Okay, so here, there is no obvious target population because of the fragmentation in the United States healthcare system. So instead, what was done is retrospective deterministic linkage with Medicare data during the period the trial is done. And so that's limited to people over 65. And 76% of the trial participants were successfully linked. And there's a reason this is pretty successful that I'll discuss in a second. But basically, even there, limited to over 65, so you're already conditioning on age, you have some sizable differences in the covariate distribution between trial participants and non-participants. And so the trial participants are younger, they have less hypertension, and you can see some of these are pretty strong differences. So these are inverse probability weighting type of results, and it's for the trial, the target result, the target population, that's the Medicare population, and then the difference of the two. So the difference between these two numbers. Excuse me. And so you can see there are meaningful differences in the treatment effect. Excuse me. The treatment effect between the trial and the target, with the treatment effect actually appearing larger after transportability. And so I just want to highlight some challenges and opportunities and one concluding slide. The first thing I want to say is in the United States, with the healthcare system we're given, defining and sampling target populations can be much more challenging than it appears. Device trials offer unique opportunities for this kind of retrospective linkage-based transportability analysis. And the reason I'm told is we know when the device is implanted, and we know the hospital in which it is implanted, so that narrows down quite a bit the set of people with whom you can link the trial data. Still, there is missing critical data that we need to define eligibility and to support exchangeability between participants and non-participants. And just as examples, ejection fraction for finely tailored risk models are not available in claims data, and lots of similar missing data issues with lab values, imaging, smoking behaviors, and so on. So lots of challenges that the registry relieves. But the big picture, I want to argue, is that as we face complex research questions about real-world target populations, we require creative use of data from diverse sources. And this will not be a binary choice. It will be some combination of trials, routinely collected data, registries, prospective observational studies, what have you, whatever it takes to answer the question. And I think the choice of data sources and causal or statistical approaches for combining information should prove a fruitful area for research going forward. Yeah, this yeah. So the question is, if we see different treatment effect results and we believe them, this suggests that there are subsets of the trial that have differential benefit. And so why would we care about the average number? I'm stating it a little more sharply. That's okay. And so I would say there's two things here. The first one is some decisions are made at group level, coverage decisions, very big funding decisions. And so you need some kind of average to estimate the treatment effect. One example might be preventive vaccination programs. Those usually are not very tailored to individuals, or maybe large-scale cancer screening programs. Those are applied at the population level. The second thing is, in general, we would expect heterogeneity over many variables on many different scales for the treatment effect. So I take heterogeneity not as something to look for in the data, but something I believe is there. So what I think is the more interesting next question for this methodology is, which is the decision relevant heterogeneity to be used. And so I think very similar methodology will be needed, where, for example, you're trying to find the optimal treatment regime for the target population, not for the trial population, but you're unwilling to or unable to use certain variables to guide that decision. So the optimal treatment identified over a subset of the potential modifiers, if it is to be identified in the target, requires very similar methodology. So I completely agree there is a tension between seeing a difference due to heterogeneity and focusing on an average effect, and we and others are kind of taking this next step analytically now. It's a good question. I mean, you could ask after.
Yeah, exactly. This is more fun.
Squirming.
I think, so can I ask this? I'm obviously a believer, but I'm going to ask a skeptical question. The Swedish trial platform is extremely important to cardiologists because of the speed and with which they're able to ask these questions across an entire country. But I worry that in that process of expediency, we're losing something really important, because nothing about the US healthcare system and the population we treat looks like the Swedish population. And so how do we understand how we would weigh a very somewhat more accurate estimate coming out of Sweetheart or Sweet PAD and a more noisy estimate coming out of Medicare data?
It's an interesting question. I think, sometimes I joke and say, if one were to run a trial in just New York or Texas, the diversity of the population would probably, which is just one city or one state, the diversity would be massively bigger than you would see in the Swedish data. And so, I think the American solution to the problem of transportability is going to be uniquely American. So I think there will be some room for routinely collected data or registries, some of which exist in cardiovascular research, of course, but even newer ones with more careful collection of baseline information that may modify the treatment effects. And then the need for transportability never goes away. It's a separate need, a separate research question from what happened in the trial itself. And so it's not just Sweetheart in the US, but a study run in the United States will often show very strong selection. So I feel the problem doesn't go away. It just the matching between the trial and the target requires some thought that is tailored to the research question.
Thank you.